All right, so welcome to part one of photosynthesis. In this lecture, we will cover chapters 8.1 to 8.3, which deals with the overview of photosynthesis, the discovery of photosynthetic processes, and then pigments that are involved in photosynthesis. Now, basically, every oxygen molecule that we breathe in was once part of a water molecule that was liberated by photosynthesis. So we can take cyanobacteria that evolved over millions of years ago and doing this process. Now, remember, I have mentioned this before, you can trace all energy back to the sun. So we use the sun either directly or indirectly for energy. And photosynthesis is one way to take sunlight and transform that radiant energy into chemical energy that we can use. So you can see on this diagram below, we have an overview of photosynthesis where light hits chlorophyll, water enters the plant, and takes in CO2, which is going to either convert sugars or give off oxygen into the air that we breathe. Okay, so photosynthesis combines carbon dioxide and water, and the end result is glucose, a sugar, and oxygen. In different organisms, photosynthesis can occur in a variety of ways. Anoxygenic, which means that it does not produce oxygen as a byproduct. And this occurs in four different organisms: purple bacteria, green sulfur bacteria, green non-sulfur bacteria, and heliobacteria. And so the helio means it's a spiral, so the ones that you see that are in a spiral are your heliobacteria. The other variety or form of photosynthesis is oxygenetic, which means that it does produce oxygen. So this is our cyanobacteria, 7 groups of algae, and then essentially all land plants that we know. In most cases, photosynthesis takes place in the leaves of chloroplast, and so we can break photosynthesis, which is kind of a lengthy process, down into 3 stages. 1, it captures energy from sunlight. 2, it uses energy to make ATP and then to reduce NAD+, which is an energy carrier that we will learn more about, into NADPH. And then 3, using ATP and NADPH to power the synthesis of organic molecules from CO2 in the air. Now, I boxed a few things here. Yellow means that it's a light-dependent reaction, which means that it does need light to carry out. And then green means that it is a light-independent reaction or carbon fixation. It occurs whether you have light or not, but it is dependent on ATP and NADPH. Now, I just realized that my notes here, I didn't put 1 or 2 for the first bullet point, so just make a note of that. I don't know what I was thinking. It's been a long day. So a simple equation of photosynthesis where we have 6 CO2 or carbon dioxide molecules combined with 6 molecules of water with a little bit of sunlight, and the end result should be sugar, C6H12O6, this is glucose, plus oxygen, which is 6 molecules of oxygen. So this is the photosynthesis equation. You can bet your dollar that this will be on the test, so make sure you memorize this equation. Now, in plants, photosynthesis does take place in the chloroplasts. So the chloroplasts are kind of like equivalent to mitochondria. They both have a double membrane, but there are some different structures that we need to learn about here. And so we will start with the thylakoid membrane. It's an internal membrane of the chloroplast. It's a continuous phospholipid bilayer, and it's organized into these flattened stacks. Now, this is where you are going to find chlorophyll, which is the pigment that captures light energy. This is also where you'll find your photosystems, which we will discuss later on in this chapter. But photosystems are basically just photosynthetic pigments clustered together. And this is also where you will find the machinery to make ATP. Now, when you get a bunch of thylakoids stacked on top of each other in columns, then you produce something called grana. So in this diagram here, you can see that we have a plant cell, and then we're going to zoom in on the chloroplast, and then notice the stacked sacs. So we have a bunch of thylakoids and producing a grana. And so you can see the thylakoid membrane, the thylakoid space, and some keywords here. Here's a much better diagram showing the chloroplast and then the breakdown of some keywords here. So on the bottom right-hand panel, you can see the thylakoids producing your granum. And then it kind of shows you a real-life picture, a scan and electron picture of a chloroplast. And then on the previous diagram, you could also see the stroma, which is a semi-liquid substance that surrounds the thylakoid membrane. And in the stroma houses all the enzymes that are going to assemble our organic molecules from carbon dioxide. And it uses energy from ATP and the reduction of NADPH So we'll talk again more in detail of these structures, but just an overview of photosynthesis. Here's a definition of the photosystems. Each pigment molecule within it is capable of capturing photons. So basically any pigment that can capture photons, and photons are packets of energy. And so light must have the proper wavelength, strikes the pigment molecule, an electron in that pigment molecule gets excited, passes that energy on to another pigment molecule. Now, this is not a physical transfer, it's just one electron exciting another electron. And so it kind of passes this on and arrives at the chlorophyll molecule, which is in the center, and just kind of starts a bunch of reactions that results in ATP and NADPH production. So here are a diagram of photosystems. The plants in photosynthesis have two photosystems, 1 and 2, and this is just a preview picture, so just look at it. Notice that it's kind of similar to our ETC in cellular respiration, where we have a bunch of proteins all lined up together and electrons are being passed through it. So it hits the first complex and it gets carried on by plastoquinone to the B6F complex. Then it gets picked up by plastocyan to another protein, and then it jumps up again. And so it's kind of this handing off of electrons or energy from one protein to another. And as a result, we are generating NADPH here. Okay, so how did we know all of this happens inside of a plant? So this is going to be story time, or the history of photosynthesis. So plants do not increase mass from soil and water alone, and so Greeks used to think that plants obtained their food from soil, and this is how plants grew. Well, Jan Baptista van Helmont, who was a Belgian doctor, decided to test this. So he planted a small willow tree in a pot of soil, and he weighed the tree and the soil before. Then he let the tree grow in the pot for 5 years, and he only added water to it. And at the end of his experiment, the weight of the tree was 74.4 kilograms, but the soil in the pot weighed only 57 grams less. So how in the world could the plant obtain food from the soil and achieve 74.4 kilograms? Okay, so soil and pot, they only lost 57 grams. So therefore, plants do not obtain food from soil. But there was a problem. He didn't really account for the water. So he incorrectly concluded that water increased the mass of the plant. So here's an overview. What do plants need? He said, okay, they need soil, they need light, they need water, they need air. What are plants made up of? Is it soil? So he tested this question, do plants eat dirt? And so you can see the diagram of the small willow tree before his experiment and after, and realized that no, plants do not eat dirt. But then he said, but they do drink water, and that's how this plant or this tree gained mass. Now I inserted a link at the bottom here. Stuart McMillan, awesome artist, takes science concepts and puts them into comic strips. And so if you are just interested in this experiment, I definitely encourage you to check it out on your own. The story continues with Joseph Priestley, who was an English scientist in the 1700s. And what Priestley did is he put a sprig of mint into the air in which a wax candle had burnt out. And your book gives you the specific date of August 17th. Then 10 days later, he found that another candle could be burned in the same air. And he was like, well, why is this? Somehow vegetation restored the air because, fire needs air in order to light or ignite. So then he decided to put a mouse in this experiment to see if a mouse could restore the air, and no, it didn't. It actually died, and you'll see that on the next slide here. So he concluded that living vegetation added something to the air, which allowed a candle to be burnt again. So you can see at the top panel pictures, he has a candle in this vase, and after a short time, the candle would die out because there's no oxygen. So then he put a sprig of mint in it, and the candle stayed lit for several days. So then he took a mouse, put it in there to see if the mouse could restore oxygen. Nope, the mouse died for science. So it was good for a good cause, I guess. So then to continue on with this experiment, he put the sprig of mint with the mouse, and the mouse was okay. So therefore, he concluded that vegetation somehow restored something in the atmosphere. So now a new question is, how does vegetation actually restore air? What's the process behind this? So Jan Ingenhousz, who was a Dutch physician, demonstrated that air was restored only in the presence of sunlight and could only be from the plant's green leaves, not the roots. So he proposed that green plants use the sunlight to split carbon dioxide into carbon and oxygen, and he suggested that the oxygen that was released as gas, and the carbon that was left over formed carbohydrates. So then by the 19th century, they determined that the overall process for photosynthesis was CO2 plus water plus light energy yields CH2O plus oxygen. Now this is an empirical formula for sugar. If you put a 2 or 3 out in front, you can pretty much form any sugar out there. So another diagram demonstrating that the plants have to be green. So notice the mint sprig. After 7 days, the candles stay lit. In the dark, it didn't work so well. And then he put a potato in there, and a potato is part of a root, so there's no chlorophyll. It's not green, and obviously that experiment did not work either. Okay, so photosynthesis includes both light-dependent and light-independent reactions. So we are continuing on our story with a plant physiologist named F.F. Blackman, and he concluded that photosynthesis is a multi-stage process with one portion that uses light directly and the other portion not dependent on light. So what he did is he measured the effects of different light intensities as well as CO2 concentrations and temperatures on photosynthesis. And he realized that as long as the light intensity was low, he could increase the light, but not the temperature or the CO2 concentration, and the rate of photosynthesis increased. But with high light intensity, if he increased the temperature or the CO2 concentration, photosynthesis also increased. So from this, Blackman concluded that the initial set of reactions depended on light, and that the second set he called dark reactions. And so dark means that it goes on whether or not there's light or not. Light could still be there and it could still carry out these reactions, but it was limited by the levels of CO2. So he found that as you increase temperature, you could also increase the rate of light independent reactions, but only to a certain point, and this is kind of like the optimal temperature, 35 degrees Celsius. Anything higher than that denatured the enzymes that carried out light-independent reactions, and then your rates of photosynthesis drastically decreased. So in this diagram, this is actually taken from his science research paper. You can see that the dark limited phase means that because there was low levels of CO2, rates of photosynthesis drastically decrease. So dark reactions are influenced by CO2 concentrations. Dark limited phase, if you have a lot of CO2, your rates of photosynthesis increases. Notice the temperature is 20 degrees Celsius. So then what he did is he jacked up the temperature to 35 degrees Celsius and realized that photosynthesis increased even more with excess CO2. Now this is dark reactions here. All right, so the story continues with oxygen and how it comes from water and not from CO2. And so the story picks up with CB, I think his name was Cass B. Van Niel, studied purple sulfur bacteria as a student at grad school. And so purple sulfur bacteria, PSB, it does not release oxygen. Instead, it converts hydrogen sulfide into pure sulfur. So here's the equation. Carbon dioxide plus hydrogen sulfide plus light energy yields a sugar plus water plus free sulfide. Now, Van Niel proposed a generalized equation where if you just replace sulfide with A, so he's got this brand new equation, replace the S with A, H2A serves as an electron donor, product A comes from split in A. So therefore, oxygen could be produced by green plants resulting from the splitting of water, not CO2, because water gets split here. So how they concluded that the oxygen comes from water rather than CO2, they studied the isotopes of oxygen. So they gave plants isotope labeled water and unlabeled CO2, and the results was that oxygen that was released was labeled. And then in experiment 2, to make sure that it wasn't CO2, they gave plants isotope labeled carbon dioxide and unlabeled water, and as a result, the oxygen that was released was unlabeled. And so oxygen does not come from the CO2, which had those oxygens labeled. So really, really cool experiment that they could do in the 1950s. I would also like to mention that on the previous slide, Van Niel just looks like a total BA with that cigarette in his mouth. But cigarette's really bad for you, so don't smoke. But I'm just saying. I mean, yeah, okay, moving on. It's way too late. All right, so the story continues looking at ATP and NADPH from light-dependent reactions and how it reduces carbon dioxide to make glucose. So Van Niel, because of his success in the previous experiment, he further proposed that the hydrogen ions and electrons generated by splitting water were used to convert that carbon dioxide into an organic matter, and he called this carbon fixation. And we will dive into carbon fixation more later on in this chapter. But Robin Neal picked up where Van Niel left off and proved that Van Niel was correct. And he demonstrated that light energy could be harvested and used in the reduction reaction, and that chloroplasts that were isolated from leaf cells were able to reduce and release oxygen in response to light. So lots of scientists involved in the process of photosynthesis, but we are not done yet. Wait, there's more. More experiments proved that electrons released from water splitting were actually transferred to NADP+, which is an electron carrier. And another experiment also concluded that chloroplasts that were deprived of CO2 accumulated ATP. If you have carbon dioxide introduced to chloroplasts, then neither ATP nor NADPH accumulated. And so from this, they knew that CO2 was assimilated or broken down or formed with other carbon molecules to form our organic molecules, which is sugar. Now, probably at this point, you're just like, okay, Mrs. E, who cares about these experiments? Well, there's three really important things from all these experiments, especially the last couple. One is that photosynthesis in plants occurs within the chloroplast. So now we have a location of where this process takes place. Two, it showed that light-dependent reactions use light energy to reduce NADP+. And remember, reduce means to gain an electron, so then it becomes NADPH, and to also make ATP. And then three, it's going to use that ATP that was made, as well as that electron carrier, NADPH, from the early stage of photosynthesis, to be used later on in the second half of photosynthesis to reduce carbon dioxide and form our simple sugars. So three really important conclusions from basically all these experiments. Okay, so the last section I will cover today is pigments, section 8.3. And so plants utilize energy from the sunlight, so they need a way to absorb this energy, and that way is through the use of pigments. Pigments are molecules that absorb light energy in the visible range, and I just want to stress that the color we see is not the color that is absorbed. It is actually the color that is reflected. So you can see in this diagram here, sunlight, which gives off visible light, and visible light is white light, hits the apple, and the apple absorbs all the colors except for the red spectrum and reflects the red, and then the red wavelength hits our eye and we perceive the apple to be red. So if you're wearing jeans, you look at your blue jeans and notice that they're blue, and that's because it's absorbing all the wavelengths except for that blue spectra color that you see. So very cool. So light is a form of energy, and it does behave as a wave as well as a particle. Light is a part of the electromagnetic spectrum, and the visible light is only a very, very small part of this massive spectrum. So if you remember from physical science, radio waves, microwaves, infrared waves, visible light, X-rays, gamma rays, I missed one in there, ultraviolet. Those are all part of the electromagnetic spectrum. So we can take visible light and we can divide it into its different colors using a prism. So you may have done that in Physical Science 9, or at least I hope you did. But because light is a form of energy and it kind of behaves as a particle, we call it a photon. So a photon is a particle of light that acts as a discrete bundle of energy. So this is where it gets confusing. We use the wave concept of light to understand different colors of light, but then we also use the particle nature of light to understand how energy can be transferred during photosynthesis. So on this top panel with the pink background, I have the electromagnetic spectrum listed from lowest photon energy to highest photon energy. So it goes radio waves, microwaves, infrared light spectrum, ultraviolet, X-rays, and gamma rays. And the spectrum that is the visible portion is the light spectrum, and it ranges from red to violet. So Roy G. Biv plays a role here. Now, my red photon is a lower energy photon energy, so you can see that it has a longer wavelength, and my blue photon has a shorter wavelength, therefore a higher energy. So if you're wondering what the heck a photon is, it's a piece of energy, has no mass, moves at the speed of light, acts like a particle, has momentum, and can be quantized, which means it can have different amounts of energy. And then a little comic relief here. Photon checks into a hotel and is asked if he needs any help with his luggage. He says, "No, I'm traveling light." So I just mentioned this previously on the previous slide. The energy content of a photon is inversely proportional to the wavelength. That means if one is up, the other one is down. That's what inversely means. So if I have a short wavelength, my photons have a higher energy. And if I have a long wavelength, then my photons have a lower energy. Now, photons kind of interact in this photoelectric effect, which means that a beam of light is able to remove electrons from certain molecules and can create an electric current, because electric current is basically just electrons moving. So the photoelectric effect can occur when photons transfer energy to electrons, and the strength of this photoelectric effect does depend on the wavelength. So if you have a short wavelength, obviously that's going to be more effective since more energy is being put, or since the short wavelength has more energy, is what I meant to say. Now the energy in photons continued here. Our chloroplasts are going to act as this photoelectric device. And so what's going to happen is that these chloroplasts are going to absorb sunlight, and then they're going to transfer excited electrons to a carrier. Now this diagram doesn't have anything to do with chloroplasts. It deals with the photoelectric effect. Notice that a red light, which has lower energy, hits potassium, and no electrons are being excited or released. A green light has higher energy. Notice that it has lower energy electrons being excited, and a blue light, which has high energy, excites high energy electrons. So it can be quantized, dependent on the amount of energy that photon has. Now, because photons can exist in different energy forms, pigments have a specific absorption spectrum that they are characterized by. So when a photon hits a molecule, energy is either going to be lost as heat, or it's going to be absorbed by electrons, which means that it's going to boost them into a higher energy level. Now, the absorption kind of depends on a couple things here. How much energy it carries, so we have to look at the wavelength. Is it high or is it low? And then finally, the chemical nature of the molecule that the wavelength hits. So we call this the absorption spectrum, which is the range and efficiency of photons, typo, that it's capable of absorbing. So organisms have many different pigments out there, but in green plant photosynthetic organisms, there are two types, chlorophylls and carotenoids. So you can see this concept map, photosynthetic pigments, we have chlorophylls and carotenoids. And so we're going to talk about chlorophyll A and B, and then talk a little bit about carotenes. We won't really touch on xanthophyll. Now, chlorophylls have a very narrow range of photons that it can absorb, but there are two types of chlorophylls in plants. We have chlorophyll A, which absorbs the violet-blue spectra, and this is the main photosynthetic pigment in plants and cyanobacteria. It's also the only pigment that can act directly to convert light energy into chemical energy, so this chlorophyll is pretty important. Now, chlorophyll B is kind of like its accessory pigment or a secondary light-absorbing pigment, but it absorbs mostly red light, and it just kind of complements and adds to the light absorption of chlorophyll A, but neither absorb wavelengths between 500 and 600 nanometers. And if you notice that on the spectra, that is where we find our green colors. So that is why we look at plants and they are green, because they are reflecting this wavelength range of 500 to 600 nanometers. Pretty cool. So here's a diagram depicting many pigments that can be found in plants. So at the x-axis we have wavelength of light in nanometers. So blue has that shorter wavelength, higher energy. Red has that longer wavelength, lower energy. And then on the y-axis we have relative absorption percentage. Now if you notice, chlorophyll A is this kind of teal color, and it does absorb in the blue range, the highest in the blue range, around like 70, close to 70%, I guess, 65%. And then we have chlorophyll B, which absorbs wavelengths a little bit lower than chlorophyll A, so it complements it a little bit. So all these pigments work together to try to absorb as much of the visible light spectrum as possible to excite electrons. All right, so the structure of chlorophylls. Chlorophylls is the pigment that absorbs photons through this excitation. Oh my gosh, okay, excitation process. I still think I said that wrong. I am so tired. Okay, so basically this is analogous or very similar to the photoelectric effect, where we have these pigments that contain a complex ring structure, and we call this the porphyrin ring. And it's just basically a ring shaped with alternated single and double bonds. At the center of this ring, we have a magnesium atom. And so the photons excite electrons in this ring, and then it reaches magnesium, and then it gets really excited. And so we call this the action spectrum, which just tells you how effective all these different wavelengths are in promoting photosynthesis. So this picture on the left-hand side, we have chlorophyll, and you can see that it has this porphyrin-like ring structure with magnesium at the bottom. But then this really cool thing is, if we look at human blood, the protein hemoglobin, which carries oxygen for us, just look at the structure of hemoglobin compared to the plant chlorophyll. Look how similar they are. Isn't that just crazy? The only difference is that plant chlorophyll have that long hydrocarbon tail, and hemoglobin doesn't have a tail. There's also a little bit difference towards the bottom with some carbons, like an additional ring in the plant chlorophyll with some additional oxygens. But just kind of cool how similar they are in shape. They have a different metal atom in the center. Plant chlorophyll has a magnesium. Hemoglobin has that iron atom. Now, carotenoids are accessory pigments. Like I said, all these pigments work together to absorb as much of the light spectrum as possible. So carotenoids consist of carbon rings linked to chains with alternating single and double bonds. So just basically these really, really, really long tails. And they absorb photons with a wide range of energies, but they're just not as efficient as our chlorophylls. They assist in photosynthesis by capturing energy from light composed of wavelengths that are not efficiently absorbed by chlorophylls. So they just try to capture everything that the chlorophylls cannot catch. Now, not only do carotenoids have this function of capturing light that the chlorophylls can't, but they also have this valuable role in scavenging free radicals. So in photosynthesis, redox reactions take place, and it can be very, very destructive because it's generating all these free radicals. So these carotenoids, which I have a typo right there as you can see, act as antioxidants to lessen the damage, so it's a protective role. Another fun fact about carotenoids, beta carotene, which is a carotenoid, if you split it in two, it produces 2 molecules of vitamin A. And for those that don't know, vitamin A is essential in vertebrate vision because it produces retinol, which is a pigment used to perceive light. So if you eat a lot of things that have beta carotene in it, it may enhance your vision. So in this diagram, we can see that the carotenoids, chlorophyll B and chlorophyll A, all work to try to capture as much light as they can and bring those excited electrons to something called the reaction center to kind of start the reaction of making ATP and generating NAD pHs. On the right-hand panel, we have diagrams that I thought would be helpful in trying to explain how carotenoids can act as antioxidants. So an antioxidant is good for your body. And what it does is it gives up these electrons to free radicals, and free radicals just means that they've lost electrons and can be very damaging to your cells. So just take a look at these diagrams and try to understand that, carotenoids, yes, they are an accessory pigment, but they also help lessen the damage in cells from redox reactions. And so with that being said, this concludes this lecture on 8.1 to 8.3. Please, if you have questions, just let me know.